understand the details about the molecular mechanism of muscle contraction or excitation contraction coupling, one should know about a particular structure in the skeletal muscle that is nothing but Sarco-tubular system. This is usually coming as a short note for your university exam. Now, let us take the definition of Sarco-tubular system. Sarco-tubular system is a modified endoplasmic reticulum having subunits. What are they? They are L tubules, terminal cisterns and T tubule. The L tubule runs parallel to the myofibrils and the L tubule make two expansion on either side. It is called terminal cistern. As you see there in the diagram, the T tubules are nothing but the invagination of the sarcolemma or the muscle membrane into the deeper part of the skeletal muscles. So, sarcotubular system mainly consists of three units. What are they? A T tubule, L tubule and terminal cisterns. Now, let us see the functions of these structures. Now, let us see the functions of these subunits of sarcotubular system. As you see there, the terminal cistern, I told you they are expansions of L tubule. What do? You can see that they store the calcium which is used for the muscle contraction. Now, what is the function of L tubule? To a certain extent, during the relaxation of skeletal muscle, this calcium is being pumped back into the L tubule as well as into the terminal cistern. Now, what this T tubule function? This T tubule is acting as a corridor or a pathway for the incoming action potential into the deeper part of the skeletal muscle. You know that action potentials are originated on the sarcolemma or on the surface. They travel and finally they enter into the T tubule. And arrival of action potential in the T tubule is the first trigger for the muscle contraction. Now, let us start the story of muscle contraction or the story of excitation contraction coupling. Now, we have seen action potentials traveling through the sarcolemma. They travel into the T tubule. They reach there in the T tubule. You know that the only language by which a nerve is communicating to the skeletal muscle is through action potential. For every excitable tissue is communicating one another through the language of action potential. Now, action potential are there in the T tubule. As you see there in the animation, you can see this action potential trigger the activation of a particular protein or a receptor. We call it as dihydropyridine receptor. Can you identify that structure there? Yes, they are interlinked with another calcium channel in the terminal cistern. What is the name of that calcium channel? Yes, rhinodinin receptors. So, dihydropyridine receptor and rhinodinin receptor, they are the connecting link between the action potential and the development of muscle contraction. So, action potential trigger the dihydropyridine receptor and this dihydropyridine receptor unlock the calcium channel, the rhinodinin receptor. The stored calcium in the terminal cistern goes out. Why calcium is going out? The reason is because the concentration gradient for calcium is operating from inside to outside. This calcium which is coming out from the terminal cistern will go and bind on a specific protein of thin filament. We have seen them having three units. Can you recollect that? Yes, you said it, troponin. Which component of the troponin? It is the C component of troponin, which is the binding site for calcium. When troponin C binds with the calcium, you can see that the troponin tropomyosin complex drifted to the next groove of actin. What it results? It results the unmasking of actin molecule. They say that one calcium binds with the troponin C can expose 7 actin molecules. Thereby, 7 myosin can make cross bridge with actin. You have seen the myosin is making cross bridge with actin. Now, what this myosin does? With the help of stored energy, the myosin head will be tilted towards the tail. I told you the myosin head is provided with an enzyme activity called myosin ATPase. With the help of enzyme, the ATP molecule is cleaved into ADP and a high energy phosphate. By utilizing that energy, the myosin head is tilted towards the tail. By tilting from either side, you can see that in the animation, the thin filaments are crossing over the thick filament. This tilting of head towards the tail with the help of energy is having a particular name. You should remember that name. This is called power stroke. So, the main activity in the sarcomere that is taking place during the muscle contraction is nothing but the power stroke. Why it is called power? Because the energy is liberated by the myosin head from the ATP. With that stored energy, the myosin head is able to, in fact, drag the thin filaments over the thick filament. Now, once the cross bridge is formed and the myosin head is tilted, pulling the thin filaments on either side. This is the ultimate molecular mechanism of muscle contraction.